thanks everyone for being here this morning. And Lisa's already said my title, so I won't repeat that. But I'm going to start my story today by briefly stepping back and considering a broad historical context of human interactions with seabird and marine mammal populations, and then try to justify why, in a modern management context and conservation context, we need a better understanding of these fields of foraging ecology, biogeography, and population biology. Okay, so dating all the way back to the origins of modern humans in coastal southern Africa, there's evidence of marine resource use to supplement dietary needs and as sources of raw materials for tools. However, starting with the harvest of North Atlantic right whales by Basque whalers in the 11th century CE, this relationship entered a new phase with the harvest of seabirds and marine mammals for market commodities, such as furs, lighting oils, industrial lubricants, baleen, and feathers. So over the course of this 900-plus year history of commercial exploitation, there's been a gradual realization that oceanic living resources are not boundless, and that humans have the capacity to trigger demographic collapses, particularly in many seabird and marine mammal populations. Okay, so what you're looking at here, are sequential declines and eventual commercial extinctions of blue whales, fin whales, and sei whales in the Southern Ocean over the last century. And this spot highlights a particular vulnerability of many seabirds and marine mammal populations, which is that they evolved life history characteristics in a context of low adult mortality, either because of large body sizes, shown by these mystacete cetaceans, or by breeding on isolated islands for seabirds and many pinnipeds. So these life history characteristics make many of these populations particularly vulnerable to the introduction of novel threats from human resource extraction, or in the case of seabirds, from the introduction of novel terrestrial mammalian predators on previously isolated islands. Okay, so these gradually developing scientific insights, along with the replacement of many marine mammal and seabird products by alternative technologies and commodities, have led to a series of fundamental shifts in the relationship of people with marine mammals and seabirds over the last century. In the initial extractive phase, where the ocean resources essentially appeared boundless, the principal underlying resource use is one of maximizing yield. And this outlook, along with the growing technological lethality and expanding geographic reach of harvests, led to an accelerating series of short-lived intensive harvests, followed in many cases by demographic collapses and commercial extinctions, localized extirpations, and in a limited number of cases with species extinctions. Starting from the beginning of the 20th century, with the establishment of transnational agreements such as the International Whaling Commission and the Convention for the Protection of Migratory Birds, a principle of maximum sustainable yield was adopted, at least on paper. And more recently, in the last 30 to 40 years, with the adoption of the Marine Mammal Protection Act, the Endangered Species Act, and other pieces of environmental legislation around the world, there's been a nearly 180-degree shift to a conservation-oriented management regime, with goals of achieving stable or increasing population sizes. However, this evolution in our management philosophies has also coincided with a period of dramatic intensification of non-harvest related human activities in oceanic habitats, earning this period the label of the Anthropocene epoch. So these activities range from increases in underwater sound production and the accelerating growth of marine transportation networks, to the intensification of fishing gear deployments and the expansion of deep water energy extraction. So these activities pose a range of indirect, non-targeted, sometimes sublethal, but also potentially cumulative effects on marine mammal and seabird populations, such as acute and chronic acoustic disturbance, vessel strikes, bycatch and entanglement, and the bioaccumulation of pollutants. And also not shown here is the accelerated dispersal of invasive species between islands. So whereas managing for maximum yield required minimal ecological information, and maximum sustainable yield, at least in theory, could be estimated from catch data and implemented via harvest limits, managing for conservation in this new threat landscape requires a substantially more nuanced understanding of seabird and marine mammal ecology, biogeography, and population biology. Okay, so more specifically, why would we care about metrics such as dive depth ranges? Well, this is an important variable in evaluating relative vulnerabilities and exposures of different species to impacts that vary by depth. As a first example, sound production and sound transmission in the ocean both vary substantially by depth, and most sound production occurs up here at the surface, while sound transmission reaches an optimum here within the sound minimum or the SOFAR channel. Exposure to many pollutants also varies substantially by depth. So for example, many of the heavy metals, such as cadmium, shown in this profile plot, readily scavenge onto particles and sink out of the epipelagic zone, and then the particle remineralizes, leaving behind a concentration maxima within the mesopelagic and upper bathypelagic. It's also important to describe metrics like marine mammal dive durations and dive behavior patterns, first to understand the potential vulnerabilities of these species to decompression trauma, if normal dive behaviors are interrupted by intense sonar or seismic mapping activities, and more generally to assess the ecological vulnerabilities of different species to chronic acoustic disturbance. Understanding how much time is spent at the surface and also where different species are spatially distributed is important in evaluating the risk, it's important in evaluating the relative exposures of different cetaceans to ship strikes, and also in being able to plan vessel traffic patterns to mitigate some of these risks. And finally, empirical estimates of population biology parameters, so population abundance and trends, are critical in assessing the risk of extinction for listing decisions, and also in understanding how quickly seabird demographic patterns or demographic parameters might respond to the removal of invasive predators from islands. Okay, so the overall objective for my dissertation was to move towards a more sustainable use of ocean resources by applying rigorous scientific approaches to fill in some of these important information gaps. So I'm going to briefly step through a high-level overview of my dissertation chapters and also try to illustrate the links back to some of these important ecological and population biology metrics. Okay, so the chapters of this dissertation were divided into two sections. The first section examined the foraging ecology and biogeography of toothed whales in the Bahamas, using data from satellite tags and biopsy samples. The first chapter in this section developed novel methods of using large legacy data sets of summarized temperature data from satellite tags, and this data informed our understanding of dive depths and dive behavior patterns in 5 species of toothed whales. The second chapter examined potential uses of fatty acids in cetacean blubber samples to estimate foraging depths for species where no tag data exists, and where the only source of information might be tissue samples recovered from biopsies or from stranded individuals. The third chapter of this section integrated all this dive information and developed a systematic framework to address differences between species in dive depth, dive duration, and surface time within a context of dive physiology and foraging ecology. So in this chapter, I also looked at differences in biogeographic distribution patterns based on these differences in foraging ecology. Okay, so the second section of my dissertation used 2 state-of-the-art modeling techniques to estimate seabird population biology parameters based on an extensive time series of strip transect surveys in the Central and Eastern Pacific. The first chapter of this section, or chapter 4 overall, estimated the abundance for 2 endangered Hawaiian seabirds using spatial habitat models that were built in a generalized additive model framework. And finally, the last chapter applied Bayesian hierarchical state space models to simultaneously estimate population abundance and population trend parameters for 4 species of seabirds endemic to the California current ecosystem and the Eastern Tropical Pacific. Okay, so in the first section of my dissertation, I used satellite tags and blubber biopsy samples from 5 species of toothed whales. And our primary study species included 2 species of dolphins, the melon-headed whale and short-finned pilot whale, the monotypic sperm whale, and 2 species of beaked whales, the Blainville's and Cuvier's beaked whales. So these spaghetti lines on this plot show the spatial distribution of satellite tracks from these 5 species. And what you can see here is that these species show a high degree of overlap within this area known as the Great Bahamas Canyon. And for orientation here, we're just east of Miami across the Florida Strait. This habitat is composed of deep oceanic canyons and basins surrounded by shallow banks, which are shown here in the light blue colors, and low-lying islands shown here in the green. And this satellite tag data set was truly globally unique in terms of the sample sizes, the number of individuals, and also in particular in terms of the number of species that were tagged in a single geographic area. And this spatial overlap allowed us to make some really interesting comparisons of how these 5 species diverged in their uses of this habitat. For this section, we used 2 models of medium duration satellite tags produced by wildlife computers called a spot and a splash tag. And both tags were implanted in the connective tissue on the dorsal fins of cetaceans using 2 short subcutaneous barbs. So both tags were also transmitted telemetry information and some dive information, although the dive information came in different formats and was based on different sensors. So importantly, the older spot model of tag had only a temperature sensor, while the newer splash model carried both pressure and temperature sensors. Okay, so chapter 1 now. For the sake of time, and because this chapter is already in the published literature, I'm just going to briefly describe what we were trying to accomplish, and then highlight a few interesting new insights that we gained from this study. So the backstory for this chapter is that for approximately the last 10 years, biologists have been deploying spot tags on cetaceans as well as sharks and pinnipeds, primarily to address spatial ecology and movement behavior type questions. But this tag can also be programmed to return histograms of high-frequency temperature recordings collected and summarized in 6-hour increments. And in the past, these time at temperature data, or TAT data, have typically just been archived, and to our knowledge, no one has really tried to make use of them to infer dive behaviors. So what I tried to do in this study was to develop a better understanding of interspecific and intersex differences in dive depth and dive behavior patterns by translating these temperature scales into approximate depth distributions, or depth scales. This involved the complex process of assimilating temperature profile data, interpolating the depths of these isotherms spatially, and then validating by comparing the approximate time at depth distributions inferred from the spot tags with direct depth measurements from the splash tag data. And I'd be very happy to discuss this with you individually after my talk. One of the interesting highlights to come out of this work was the ability to look at diurnal patterns over long and relatively continuous time series. So what this is a representation, so this representation here shows the vertical habitat use of an individual melon-headed whale. And the shading of these cells indicates the proportion of time that was spent by this individual in each depth/temperature strata. What this shows is that during daytime periods, in the light colors here, this species didn't dive below 24 degrees centigrade or approximately 100 meters depth. And instead, it appears to confine all of its foraging dive activity to nighttime periods, which are shaded here in gray. We were also able to look at what proportion of time different species were spending within their approximate foraging depth ranges. So these box plots show that the 2 beaked whale species in our sample had very low ceilings on the proportion of their overall time budget that they could spend within their foraging habitats. And this is when compared with the delphinids and the sperm whale. And this raises an interesting question of why the beaked whales might tolerate only being able to spend, on average, about 27 to 28% of their time actively foraging. And I'm going to circle back to this question and try to answer this at the end of chapter 3. Okay, so moving on to chapter 2 now. The backstory for this chapter is that there remain some key gaps in our knowledge of toothed whale dive behavior, and also some important questions like three-dimensional habitat overlap that we can only really address with dive information on all of the species that are present in a community. Successfully deploying tags on all of these species is an incredibly challenging process and also is quite expensive. And so there remain several species in the assemblage in the Bahamas, including the dwarf and pygmy sperm whales and the Gervais beaked whale, that haven't successfully been instrumented yet. And there are also many more species that are untagged worldwide. So in this chapter, I examined whether we could estimate foraging dive depths for these untagged species using fatty acid relative abundance from blubber samples that can more readily be collected using widely applied punch biopsy techniques, and also from tissue samples recovered during strandings. Okay, so briefly, fatty acids are a diverse class of essential organic molecules, and the relative abundance of different fatty acids has been widely applied as a signal of prey composition. There are a few particular properties specific to different groups of fatty acids that make them useful as tracers of diet, and these include a unique biosynthetic origin in a particular group of autotroph or heterotroph, and deposition in body tissues with minimal chemical modification. So based on these properties, the polyunsaturated fatty acids, or PUFAs, represent an intriguing potential tracer of depth. So first, in a marine environment, PUFAs are almost exclusively biosynthesized by photosynthetic autotrophs up here in the euphotic zone. PUFAs are also essential fatty acids that are preferentially consumed and degraded from particular organic matter, or POM, as it sinks and is remineralized. And in combination, these source and sink dynamics are thought to underpin this observed decline in PUFA relative abundance from POM samples collected at 2 stations in Southern California by Jones et al. 2008. Marine mammals and other consumers below them in the food chain also generally lack enzymatic pathways to modify PUFA chain length and double bond structures. And so we hypothesized that these gradients in POM might also filter up through the food web to cetacean blubber. We also examined the relative abundance of odd chain length fatty acids, whether they might vary in a predictable manner with depth. And this group of fatty acids are thought to originate exclusively in heterotrophic bacteria. And Jones et al. Empirically demonstrated a strong increase in this bacterial fraction in POM samples between the surface and 100 meters, although below 100 meters the trends were much more equivocal. Okay, so the results from the polyunsaturated fatty acids in cetacean blubber samples met our expectations based on the Jones et al. Study and showed a highly significant decline in relative abundance here on the x-axis, with increasing foraging depth here on the y-axis. And this negative relationship was found using a set of individuals where we had both biopsy and tag data. But these individuals represented a fairly limited subset of our overall biopsy data set. The vast majority of samples weren't associated with a specific tag record. So we also used mean foraging dive depths characteristic of each species, and similarly found highly significant declines in that data. Overall, relative abundance of the odd chain length fatty acids did not show any significant relationship with foraging depth. However, after that negative result, I performed a subsequent exploratory data analysis to look at relationships between mean foraging depth and specific ratios of odd chain length fatty acids. And this exploratory data analysis revealed several highly significant relationships of specific ratios with foraging depth, including the ratio of the 2 most abundant odd chain length fatty acids in our samples, the saturated C15 and the monounsaturated C17. So this relationship was also highly significant using species means. So we had 12 biopsy samples from individual Gervais beaked whales, and using a mean of their PUFA relative abundance values, we predicted a foraging depth for this species of 1,102 meters based on the PUFA foraging depth model. And the model of C15 to C17, 1, and 8 agreed very closely with the PUFA depth model, yielding a predicted mean foraging depth of 1,072 meters. And this prediction falls right between the Blainvilles and Cuvier's beaked whales. And I'll circle back to the significance of that depth in terms of foraging ecology and dive physiology for the species at the end of chapter 3. Okay, so more broadly, we think that if these patterns can be validated in other species and in other habitats, that this approach has substantial potential to use existing biopsy samples and stranding samples to gain new insights into dive behaviors and foraging ecologies in many cryptic beaked whale species around the world. And also in particular for dwarf and pygmy sperm whales, which are relatively abundant in the Bahamas, but which are also shy and inconspicuous, and to date have eluded all attempts at instrumentation. Okay, so moving on to chapter 3 now. So this analysis attempted to develop a framework to better understand why these 5 deep-diving toothed whales might exhibit such divergent foraging dive depth ranges. And these ranges are shown in the gray shading on this histogram plot. To address this question, we have to start by thinking about a fundamental challenge that's faced by many cetaceans and other secondarily marine adapted animals, like seabirds and sea turtles, and that is that their prey resources, which are shown here in this acoustic backscatter plot, are distributed at various depths that are vertically separated from their oxygen sources up here at the surface. This necessitates commuting and also limits the amount of time that can be spent foraging during any given dive. So this fundamental separation imposes some important trade-offs on the foraging ecologies and biologies of toothed whales, particularly those that specialize in deep diving. On the one hand, there are ecological choices in terms of where prey is densest, where prey is least capable of evading capture, where the nutritional quality of prey is highest, and where interspecific competition is lowest. And all these properties vary strongly by depth. On the other side of the ecological trade-offs are evolutionary and biological variations in gross morphology. So for example, how large does an animal need to grow to efficiently access its prey? There are also differences in cellular physiology. So for example, how much can an animal concentrate hemoglobin and myoglobin in body tissues? And finally, there are also differences in the proportion of time allocated to different dive behaviors in the dive cycle. So for example, how much time is allocated to foraging as opposed to commuting, as opposed to recovery between dives. Okay, so our initial working hypothesis for this analysis derived from this 2000 paper by Sean Norrin and Terry Williams up at UC Santa Cruz, which described this very tight relationship between body mass here on the logged x-axis and maximum dive duration here on the logged y-axis within the toothed whales or odontocetes. The theoretical reason why dive durations would increase with body mass is that metabolic rates scale allometrically as a positive but decelerating function of body mass, while oxygen storage capacity scales isometrically as a linear function of body mass. And this creates a divergence which allows larger species to sustain longer dives. However, when we confront this hypothesis with the observed data from our tag species, so here in real number space instead of log space, we find that the Norrin and Williams model provided a reasonably good fit for the 2 delphinids and the sperm whale, but substantially underestimates the maximum dive durations of the 2 beaked whale species. And further, if we try to fit a model between observed maximum dive duration and just body mass alone, the result is a much weaker model fit. Okay, so what specifically about the beaked whale's dive behavior might allow them for these unexpectedly long dives relative to their body masses? Well, one empirical difference you can see in these time depth recorder profile plots is that the 2 species of beaked whales almost always punctuate their prolonged foraging dives with extended recovery periods or interdeep dive intervals. And these intervals consist of either surface time or short, relatively shallow bounce dives that are non-foraging in nature. This pattern contrasts markedly with the short surface intervals and back-to-back deep foraging dives exhibited by the delphinids and the sperm whale. Okay, so now including the median length of interdeep dive intervals, or IDDI, as a covariate, along with body mass, yields a much stronger explanation of the observed pattern of maximum dive duration, with explaining upwards of 93% of the variance. Okay, so what biologically do we think is going on with the beaked whales? Well, our new working hypothesis, and this is based on the work of Tyack and earlier studies by Jerry Koyman and Paul Panganis, is that the small and medium beaked whales are relying partially on muscular anaerobic respiration during dives, and thus are exceeding aerobic dive limits, or ADL, commensurate with their body sizes. The cost of exceeding ADL is that lactic acid accumulates in body tissues, and this takes a significant amount of time to aerobically reprocess. Okay, so now circling back to this plot, comparing the proportion of time different species spent within approximate vertical foraging ranges, What this shows is that because of the longer recovery periods thought to be associated with exceeding ADL, the beaked whales spend much less of their time, overall time budgets, within their foraging habitats, with a maximum of less than 35% spent in the lower mesopelagic and bathypelagic. And that compares with upwards of 70 to 90% spent by the sperm whales and delphinids. Okay, so why might the beaked whales have adopted this seemingly inefficient strategy instead of targeting prey that's more commensurate with their body sizes and aerobic dive capacities? To address this why question, let's think in general terms about the ecological context in which these animals have evolved and in which they make their foraging decisions. So first, there's generally a daytime deep scattering layer peak in prey biomass and prey density within the mesopelagic. And part of this peak migrates towards the surface at night. Below the deep scattering layer, prey biomass and density both typically decline quite precipitously, although there is often a small uptick in consumer biomass just above the benthos. Additionally, prey deeper in the water column typically relies to a greater extent on the refuge of darkness to avoid its predators, and thus exhibit decreases in streamlining and evasiveness with depth. So coinciding with these differences in evasiveness, we also typically observe increases in water content and decreases in the protein content of prey with depth. And finally, there are fewer air-breathing predators with the specialized adaptations necessary to reach those depths. So that results in generally less intense competition for prey. Okay, so why might the beaked whales have adopted this counterintuitive strategy? We think that the beaked whales exploit a less abundant but also less evasive lower mesopelagic and bathypelagic prey resources, or we hypothesize that in a relatively low competition niche, all without growing large. And we think, that smaller body masses may be particularly important in these deeper habitats, since the additional body tissues that would be necessary to access this habitat as an aerobic diver require a substantial metabolic investment. And not making this investment may allow them to sustain themselves on the basis of more calorically limited lower mesopelagic and bathypelagic food webs. On a side note, we don't have direct dive data for the Gervais beaked whale, but our fatty acid proxies place this species right here in between the Blainville's and Cuvier's beaked whales. And this suggests that an animal which weighs approximately 17% less than the Blainville's beaked whale likely faces similar challenges in accessing its bathypelagic foraging habitats without exceeding aerobic dive limits. So we would expect similar dive behavior patterns. In contrast to the beaked whales, the large body masses of sperm whales enables this species to efficiently aerobically access a wide range of depths. And in this habitat, they typically target the more abundant and comparatively higher density mesopelagic prey layers. Sorry, out of sync. We hypothesize that the extremely large body mass of sperm whales also presents drawbacks, requiring consistently large caloric inputs and limiting their ability to sustain themselves on sparser food webs above and below this mesopelagic optimum. Finally, the smaller body masses and more aerobically active dive strategies of the delphinids limit their ability to access deeper prey resources. However, these adaptations, which reflect their evolutionary origins in a predominantly epipelagic and neritic family, also allow these species to pursue more evasive and likely more energy-rich vertically migratory components of the deep scattering layers. I missed all those cues. Okay. So these familial differences in foraging depth adaptations are also factors in the response of different species to daytime and nighttime light variation. So what you're looking at here are foraging dive depth distributions during daytime and nighttime periods that have been normalized to the median depth of nighttime dives along this line. So for example, at day values that are below this line, such as this one, indicate that daytime dives that are deeper than nighttime dives, and conversely, the converse is true also. What this first panel shows is that the melon-headed whale with the shallowest dive capacity appears to entirely dependent on prey migrating up into the range of its relatively short foraging dives at night. The two intermediate divers, the short-fin pilot whale and the sperm whale, respectively dove substantially and slightly deeper during daylight periods, reflecting likely the normal diurnal vertical migration of prey. Intriguingly, the Blainville's beaked whale showed a somewhat shallower foraging dives during the day, potentially reflecting either a reverse diurnal vertical migration of prey, of a prey species, or a switching of prey species between day and night. And finally, the deepest diver in our set, the Cuvier's beaked whale, exhibited very little difference between daytime and nighttime foraging dives. So finally, these differences in dive depth ranges between species and genders also had important implications for the spatial distributions of these toothed whales species. So briefly, these can be categorized into a wide-ranging distribution pattern, typical of the delphinid species, and the subadult male sperm whales, which are these light-colored purple dots here. And we hypothesize that this reflects a use of broadly distributed deep scattering layer prey resources. This distribution pattern contrasted with the more localized distribution of the deeper diving species, so the female sperm whale, shown in the dark purple here, and also in the two beaked whales. And we hypothesize that these more localized distributions reflect a spatial association with benthic habitats that are accessible within their foraging dive depth ranges. And those are shown in this blue shaded area ringing the canyon system here. Okay, so now thinking back to the conservation questions that were raised in the introduction, what have we learned? Well, first, on the subject of dive depth ranges, I've shown methodologically that summarized temperature data and fatty acid proxies can both yield important information on dive depth ranges in toothed whale predators, and this information has the capacity to inform hypotheses about relative exposure to depth stratified sound and chemical pollutants. I've also shown that there are key interspecific differences in dive duration and dive behavior that may make some species more vulnerable ecologically to chronic acoustic disturbance from naval sonar, seismic mapping, and other ship noises. For example, the beaked whales likely subsist on a relatively marginal prey resource and also invest a greater proportion of their time into each dive, and therefore any interruption of normal foraging behaviors, as has been observed during experimental sound exposures, may have a greater impact on the stress and reproduction and ultimately survival of the beaked whales relative to other species. We've also shown that the beaked whales spend a proportionally greater amount of their time of their dive cycle in the surface waters, and that may potentially increase their vulnerability to vessel strikes. And finally, the biogeographic information presented in the last slide can begin to inform spatial management of human impacts, such as the distribution of naval training exercises and shipping lanes. Okay, so now switching gears somewhat, the next section of my dissertation explored the use of two state-of-the-art modeling techniques, so generalized additive models and Bayesian hierarchical state space models, to estimate population abundance and growth rate parameters for several small and medium procelluriform seabirds. So what are the importance of these parameters? Well, as a group, the storm petrels, and shearwaters experience particularly high rates of population endangerment, with 31 to 45% of species listed by the IUCN Red List with a status of vulnerable to critically endangered. As I alluded to in the introduction, many of these species generally lack long histories of coevolution with terrestrial mammals. And this makes many of these species particularly susceptible to the introduction of these species to islands. Species like rats, cats, stoats, and pigs. So this susceptibility is due to small and medium body sizes, extended lifespans, and low annual reproductive outputs, as well as burrow, crevice, and ground nesting habits. Because of these vulnerabilities, substantive empirical estimates of population abundance and growth rate are key parameters in accurately assessing the risk of extinction for listing decisions, and in strategically targeting limited conservation resources. However, many of the petrels, shearwaters, and storm petrels have also adopted breeding strategies, often in response to their traditional avian predators, that make it very difficult, if not impossible, to apply random sampling and rigorous statistical methods in colony-based surveys. And these challenges include cryptic burrow entrances, nocturnal or crepuscular patterns of colony attendance, and steep, inaccessible, or vegetatively challenging breeding habitats. Okay, so the rigorous sampling of seabird densities at sea using strip transect methods, which are illustrated in these two diagrams, provide opportunities to resolve some of the challenges associated with colony-based data. So first, in terms of detectability, non-diving seabirds are relatively conspicuous and thus readily counted and identified within reasonable distances from a research vessel during daylight periods. And also in terms of accessibility, oceanic habitats present relatively few barriers to research vessel access, allowing for a comparatively unbiased sampling, randomized or systematic sampling of underlying seabird density distributions. Okay, so both chapters in this section employed an extensive extensive time series of 17 seabird strip transect surveys conducted by the NOAA Southwest Fisheries Science Center, and covering an enormous swath of the eastern and central Pacific. This plot shows the distribution of those transect surveys in the eastern tropical Pacific, the central Pacific, and in the California current ecosystem. And among seabird data sets worldwide, this time series ranks as a truly globally unique data set with its vast geographic extent and also its multi-decadal duration. To give myself a little bit more time to delve into Chapter 5, I'm just going to briefly touch on Chapter 4. So in this chapter, I was trying to estimate the abundance for two endangered Hawaiian seabirds, the Newell's shearwater and Hawaiian petrel. They're both endemic to those islands. And in this analysis, we used the cumulative observations from 9 surveys over 16 years, which are shown in the red dots. They aren't showing up very well on the screen, but red dots here, to develop spatial habitat models for the Newell's shearwater using a generalized additive model algorithm. So we then used these density predictions to estimate abundance, and also estimated the uncertainty surrounding these estimates with a non-parametric bootstrap approach. So here are similar results for the Hawaiian petrel. And these results were significant to the seabird community because of the conservation status of these seabirds. But I'm not going to touch on these anymore, but would be happy to discuss these with you further after my talk. Okay, so Chapter 5. In this final chapter, I'm going to present an approach to estimate abundance, and in particular to evaluate population trends in these 4 small and medium proselyriform seabirds using Bayesian hierarchical state space models and at-sea data. As an overview, inferences about seabird abundance trends from at-sea survey data have often been elusive because of a combination of true year-to-year variation around an underlying population, or of the underlying population, as well as sampling error associated with the inherently stochastic nature of encountering seabirds during a transect survey, and also with the relatively small fraction of the ocean surface that can be effectively sampled in any given year. And this variation, which is shown in these point estimates of Townsend's shearwater abundance from 8 years of sampling in the eastern tropical Pacific, or this is shown in those points. One approach to estimating trends that's been applied previously involved a 2-step process in which abundance is first estimated within each year, and then subsequently a regression is fitted to these annual point estimates. However, this approach faces several challenges. So first, there's limited statistical power because of small sample sizes. So all the sampling information has been reduced to these 8 points. And also, uncertainty in each abundance estimate isn't propagated through to the uncertainty around the growth rate parameter R. So this approach this approach also lumps together true variation in the underlying population process with nuisance error associated with the sampling technique. So the hierarchical or mixed effects framework provides an improvement over that former method, as it allows all the data to contribute to the joint estimation of some overall parameters, such as initial abundance and some not, the growth rate parameter R. And in this model, we also include a random effect process error term, epsilon sub t, which allows for idiosyncratic year-to-year variation away from these mean parameters. Finally, developing these models in a Bayesian framework provided as outputs posterior distributions that were relatively easy to interpret from a management perspective. Okay, so now let's look at the state space component of these models, which allowed the data to be formally represented as the outcome of two or more distinct stochastic processes. So the first process that we were trying to model was the population change through time. And here we've represented this with an exponential process with some normally distributed random effect variability. However, this framework is also very flexible, so other formulations are relatively easy to implement. In this chapter, I also implemented a Markov process model, although I'm not going to show the results of that one in this talk. This underlying ecological dynamic, however, can only be observed through a sampling process which carries its own sources of stochastic error. So in this model, we've broken this error down into two components reflecting different scales of Siebert distribution. The first observation process that we're trying to model describes the rate at which observers encountered groups of one or more seabirds in the survey strip, given some underlying seabird density distribution. And we've represented this process as a generalized Poisson distribution with units of encounters per square kilometer of survey area. And in this formulation, it allows for the relaxation of the strict Poisson equality of mean and variance. And that accommodates for large counts that result from heterogeneous spatial distribution of seabird foraging habitats. The second observation process that we're trying to model describes the number of birds comprising each group. And this we've represented as a truncated negative binomial distribution. And this allows for overdispersion to accommodate for occasional elevated counts resulting from seabird aggregation behaviors around small-scale foraging features like flocks. Okay, so now putting it all together, here's the overall model framework. And we've added a little bit of additional complexity here because of the effect of bird movement relative to the survey vessel, and also the proportion of the population represented in different stratum within our overall study area. Okay, so overall, we think that this approach of differentiating a biological state process from a nuisance observation process provides a more explicit, mechanistic, and ultimately interpretable description of how the data were actually generated. Okay, so we've applied this model framework in 4 initial case study species: the IUCN critically endangered Townsend's shearwater, the near-threatened black-vented shearwater, the endangered ashy storm petrel, and the least concern black storm petrel. These species were mainly selected because they represent breeding resident populations, and upwards of 95% of their entire at-sea ranges are contained within our study area, or our study areas, plural. This allowed us to minimize the influence of emigration and immigration on the population growth rate parameter R, and allowed us to make inferences about actual population change through time. After applying the exponential version of the Bayesian hierarchical state space model, here are the estimated posterior distributions of annual abundances and the median trend for these critically endangered Townsend's shearwaters. And the uncertainty as to whether this population has actually declined is also represented in this histogram plot, which shows the posterior distribution of the growth rate parameter R. 82% of this distribution falls below 0, which is this vertical line here. And that indicates a better than 50-50 chance that this population has indeed declined from a median initial abundance of approximately 83,000 individuals in 1987. Okay, so here are some of the same plots for the black-vented shearwater nesting off the Pacific coast of Mexico. And this plot shows a fairly strong evidence of an increase with less than 0.7% of the posterior distribution falling below 0. Inferences for the black storm petrel and the ashy storm petrel showed much greater uncertainty. However, both species did show modest evidence of increasing trends. So for example, the ashy storm petrel here, 82% of the posterior fell above 0. Okay, so what have we learned from this modeling exercise? Well, to frame this discussion, I'd like to start by quoting the statistician George E.P. Box, that essentially all models are wrong, but some are useful. And I think that provides a useful perspective, or helpful perspective on this modeling exercise. We don't think that the specifics of model configuration and parameter estimation in these case study species provides an exact description of a much more complex underlying reality of population processes and also sampling variability. However, this exercise shows that the Bayesian hierarchical state space model approach using ATC data can capture a sufficient portion of the underlying dynamics of population change and sampling design to yield useful information for conservation management of these species. Although there is considerable posterior uncertainty surrounding each of these annual abundance estimates, these estimates provided the first range-wide estimates of population abundance for many of these species. And also the trend and abundance estimates generated by these models and ATC data generally agreed with the IUCN listing decisions, which are shown in these red ovals. Finally, I'd like to also highlight here that this limited set of case study species, I think, points to the importance of invasive predator eradication on islands as a key management tool. So these 3 species in this study showed the 3 species that showed some indication of population increases all breed over at least a portion of their range on islands where invasive predators have been eradicated in recent decades. By contrast, the Townsend shearwater, which exhibited a declining trend over our study period, breeds on Socorro Island, on which to date feral cats persist. Okay, so that's all for my chapters. And I think I've run over by a bit. So I just wanted to briefly thank the many people who've been integral to the creation of these data sets, including our co-PIs, our field collaborators, and the many funders of my graduate research and also of these amazing projects. I've also had the really great fortune to work with some amazing and talented people throughout my graduate school career. So Lisa Valance and Paul Dayton have been just amazing supporters and mentors and have challenged my thinking and made me a much better scientist. So really, thank you very much, Lisa and Paul. I really appreciate it. I also want to thank my committee, Bryce Simmons, Lynn Talley, John Turin, and Carolyn Curley for their feedback and support through this process. And I'd like to give a huge thanks to our SWAG lab group up at the Southwest Center and to our 2010 cohort, and especially to Rachel Morrison, who we really miss. I also would like to give a heartfelt thanks to my family, and especially to my mom, Lynn Shepard, who's helped to nurture me all along this path. And finally, to my wife, thank you so much for all your love and support and kindness and over many years of working nights and shortened weekends and months of long absence. So, all right, any questions?